My name's Gerry Feeney, as you've just heard. What I wanted to talk to you today was about my experience being a patient on a very early stage clinical trial, and also the benefit of having had whole genome sequencing to signpost other trials and other potential therapies. So a quick overview of my talk today. I'm going to have a quick overview of my sarcoma journey. I think this may resonate with some of you who've had similar experiences. I'll explain the trial, how I was recruited, the trial procedure, the outcomes, in particular how outcomes of such a trial are assessed, and other treatment options which became apparent as a result of my whole genome sequencing. So my sarcoma journey, and some of you may relate to this from your own experiences, some of which we've heard today. So I first presented back in late 2017, very odd lump on my left leg. This was initially misdiagnosed by the GP as liposarcoma, then as a cyst, but I was referred to the Nuffield Orthopaedic Centre in Oxford, and here I was diagnosed initially with angiosarcoma, then undifferentiated pleomorphic sarcoma, and finally landed on myxofibrosarcoma. And I think this goes to show how difficult it can be to diagnose the sarcoma subtype, but that's actually important because the subtype can inform the treatment. So along the way, I've had 2 courses of neoadjuvant radiotherapy. That's radiotherapy before the operation. I've had 6 operations with plastic reconstructive surgery. I actually had a vascular graft as well, and a reconstructed artery, vascular reconstruction. There's been a number of complications after surgery. I've had numerous infections. I even had maggots in the wound at one point during the hot summer during COVID. I had cellulitis which escalated to sepsis. I've had pulmonary embolisms in both lungs after one operation. I've been left with some residual mobility impairment. I've got lymphedema in my leg. I've also got a proprioception disorder, which has led to some embarrassing falls, hence the crutch. But however, I'm fortunate not to have had lung metastases after so many recurrences, and I'm told that's unusual. Nevertheless, I had a further recurrence this year. I've now got 2 active tumours in my left leg. The surgeons at Oxford said it's really time to amputate now for local control to stop it spreading. I was therefore referred to the Royal Marsden for, as a last resort, for an isolated limb perfusion. And this is quite interesting because what they do is they tourniquet the top of your leg and they circulate strong chemo through your leg, which doesn't go systemically, so you don't get the liver and kidney debilitation. I think you mentioned doxorubicin can be quite debilitating. However, after extensive assessment, I was considered unsuitable for the ILP because I've had so many operations on my leg, the circulation wasn't up to it. And also the type of chemo they use in an ILP, which is melphalan and TNF-alpha, it doesn't get a good response from myxofibrosarcoma. So I was referred on to the trials unit. Unfortunately, they were recruiting for a trial of an oncolytic virus. And this is very interesting because it's a bioengineered, a synthetic virus. The idea is that it attacks cancer cells, but it doesn't hurt healthy cells. And also, interestingly, it creates a vaccine effect which prevents spread. Now, this is an open label. It was registered in the US and the UK. It's an open label trial. And what that means is no blinding, no randomization, and all patients get the same treatment. It's a very early stage phase 1 trial. I was only the 3rd human to have it after the mouse model in the lab. The trial is split into 2 phases. It's phase 1, so it's an early stage trial, but phase 1A is the oncolytic virus drug, which is called Ixovex-1, with on its own as a monotherapy. Phase 1B is the oncolytic virus together with immunotherapy. Now, I actually think this is very interesting for sarcoma patients, and I'll come on to that in a little while. It was a commercial trial sponsored by the pharma, so they paid for everything, all the hospital time. It wasn't an academic trial sponsored by the charities. So here we have Ixovex-1 is the oncolytic virus drug. The indications it's meant to treat is palpable tumours. And by that, it means tumours they can actually see and feel and inject. And I have 2 tumours, one of which is palpable. It was through the proof of concept, through the mouse model, the preclinical in the lab, and now it's in phase 1. Now, the objective for any phase 1 trial is toxicity. It's understanding what the side effects are. The next step would be phase 2, and there it's about, does it work? How effective is it? And the next step is phase 3. Is it better than what we already have? And there may also be a phase 4 where they look into what are the long-term effects of the treatment. Now, the trial procedure itself, you can see here a screenshot of the My Marsden app, which is a really good app the Royal Marsden use to tell patients when they're having appointments coming up, blood test results, any letters and so forth. Now, before I went on the trial, there was quite an extensive qualification phase, and this is to make sure that I complied with the inclusion and the exclusion criteria. Now, as you can see, this is way more intensive than you normally have pre-op for a medical intervention. And the reason for that is because it's a trial, such an early stage trial, they have to make sure they really understand your comorbidities and everything else about you to make sure you meet the criteria. Then during the trial itself, the level of assessment and testing was even more intensive. I mean, I had 18 blood tests over the period of 6 weeks. So they're really wanting to look and see what are the side effects of this treatment. What they also wanted to know, though, was, is it starting to work? So I started to have ultrasounds to measure the size of the 2 tumours while I was having the treatment. So the actual treatment itself took place in 4 cycles. So each time I'd have an injection in the tumour, and they'd keep me in overnight to see what the side effects were and undergoing tests. And in fact, there were a really good dedicated team at the Royal Marsden. The principal investigator, Professor Harrington, he wanted to be phoned at 3 in the morning, the night of my first cycle, to see how I was getting on. So they're very dedicated, an excellent team there. Now, what they did was, for the first 2 cycles, I had a half a dose just to see how I was reacting. And then for the last 2 cycles, it went up to a full dose. And that is what they expected to be the maximum tolerated dose before going on to phase 1B. Now, I finished the trial, but I don't know the results yet. So the next step will be really, they're still looking to see if there's any long-term side effects, but now they're looking to see, has it worked? So I've got an ultrasound, a biopsy, and a CT scan coming up at the end of this month, on the 25th. Now, trial outcomes. So there's 2 aspects to trial outcomes. One is the patient-reported outcomes. That's what I reported having experienced, as well as toxicity. And what they expected was, after they injected me with a virus, it's a synthetic virus, but it's still a virus, and so it creates a rigor. And what a rigor is uncontrollable shaking. Now, I've had sepsis before, so when you're getting an infection coming on, you get this uncontrollable shaking. And that happened about 6 hours after the injection. That was followed by a fever, but it all passed overnight. And then I had some other side effects over the next few days, which were due to mild liver toxicity from the treatment. But the other bit is tumour response. So has it actually worked? And there's 2 aspects to this here, is measurement of size and measurement of tumour activity. So for measurement of size, they use a very structured scale called RECIST. That's response evaluation criteria in solid tumours. And what they're looking to see here is it complete response, partial response, stable disease, or progressive disease? And they figure this out by taking the sum of the longest diameters of the tumours. And I've got 2 tumours. So this is how they'll assess the trial outcome, but I won't know that yet till the end of the month. However, yes. Now, phase 1B. This is interesting for sarcoma patients. They haven't started this yet. It's starting in January. Now, what I should explain here is that tumours responsive to immunotherapy are known as hot tumours, and tumours that don't respond to immunotherapy are known as cold tumours. Now, there's something called the tumour mutational burden, the TMB, and that's seen as a proxy indicator for whether tumours are hot and cold. A high TMB implies sensitivity and responsivity to immunotherapy, and the immunotherapy we're talking about here is the immune checkpoint inhibitors. That's the anti-PD-1, PD-L1. I'll say a bit more about that in a second. But these immune checkpoint inhibitors allow the immune system to attack and destroy the cancer cells. Now, sarcomas are seen as cold tumours. They have a low TMB and typically not known to be responsive to immunotherapy. But the oncolytic virus is intended to alter the tumour characteristics to make them respond to immunotherapy. So let me just show you something here now. So if the immunotherapy that they're planning to use in phase 1B is a PD-1 inhibitor, a PD-1 is a programmed cell death 1. It's a protein. It acts as a brake receptor on T cells or immune cells. Now, if that protein is inhibited, if PD-1 is inhibited or suppressed, it allows the immune cells or the T cells to see and attack the virus-altered cancer cell. Now, if you look at TMB, tumour mutational burden, typically, if it's greater than 10, it's seen as a hot tumour. If it's lower than 10, it's seen as a cold tumour. My understanding is that the NHS wouldn't prescribe immunotherapy for a tumour with a TMB of less than 10 because they don't expect a good response. Now, my TMB as a sarcoma patient, which I found out as a result of whole genome sequencing, is only 2. So this is typical of sarcoma tumours, I understand. So the interesting aspect of phase 1B is that it could mean that with the benefit of something like an oncolytic virus, sarcoma tumours could actually be treatable or more treatable with immunotherapy. Now, I mentioned I may have other options because of whole genome sequencing. So here, when I was treated at Oxford, I had the Oxford Precision Oncology for Sarcoma, the OXPOS analysis. And this has 2 benefits. First benefit is it tests your germline predisposition or potential predisposition to cancer. So I think the question came up earlier is, could your kids have cancer? Could your brothers and sisters have a predisposition to cancer? And this can basically identify, do you have a germline vulnerability to cancer? The other thing is genomic profile of the tumour itself. So what it can actually do is it can signpost potential treatments. And what it told me was that I have actually a BRCA2 gene mutation. And as a result of the whole genome sequencing, it signposted 3 potential treatments, which had hitherto not been identified. The other good thing about this precision oncology, OXPOS, using whole genome sequencing is it doesn't just identify potential chemotherapy treatments that may work for you, it also identifies from your tumour whether or not it's radiotherapy sensitive. In other words, how effective radiotherapy is going to be. So now here, what I'd like to just do is to refer to some really interesting training I had last year, the European Organisation for Research into Treatment of Cancer, Patient Days. A very interesting lecture from a doctor from Birmingham University, who's basically saying if we understand the genetic mutations that are causing cancer, regardless of what sort of cancer it is, it can actually signpost the treatment. So some basic principles of genome sequencing here. Tissues are made of cells. Each cell contains 46 chromosomes, and chromosomes are made up of DNA. Now, if we look, the DNA is like the books in a library, and the genome is your library, it defines you. And cancer is the result of a DNA aberration or mutation. And whole genome sequencing, that can identify any impactful mutations, which can be likened to misspelt or repeated words or sentences in your DNA books. So let's now go to my OXPOS report. My OXPOS report basically says that they've identified I've got a BRCA2 mutation. It's called MZ amplification, and it signposts potential sensitivity to platinum chemo and also PARP inhibitors. So if we look here back to the patient days training, we can see that actually MZ amplification is a kind of chromosomal instability. And for this, DNA replication inhibitors are signposted for chromosomal and genomic instability. And the 2 things they signpost are cisplatin, which is platinum chemo, and also PARP inhibitors such as olaparib. Now I should explain that PARP is an enzyme that allows cells with damaged DNA to repair themselves. But healthy cells have got other repair pathways. cancer cells don't. So if you actually if you inhibit HART, the enzyme, the cancer cells should die off. But my OxPOS not only did it identify these two treatments, cisplatin will be standard of care, olaparib will be off-label for my indication, but still possible to be prescribed. But the OxPOS analysis also identified another treatment, which is an immunotherapy under development called stimulator of interferon genes, STING agonists for short. So you can see here, that's 3 genome-guided therapy options, which were hitherto unidentified for me. So, you know, I'm pretty attached to this leg after all we've been through. So an amputation postponed could be an amputation avoided with these treatments. Now, I've talked a bit about trials, and you may wonder, how do I get on a trial? I don't think your MDT would refer you for a trial if there was a standard of care option available. But a nurse once said to me, the NHS is there for you, but sometimes you have to be opinionated and influence and manage your pathway. And so I've always found it's useful to understand the disease, and in particular, to understand the upcoming treatments. And these 3 websites I found very useful. Sarcoma UK's Clinical Trials Hub, which is trials for sarcoma patients. The National Institute of Healthcare Research has a website for all the trials in the UK called Be Part of Research. And clinicaltrials.gov is an American website, US website. It's a global registry of trials. But all these trials need to be funded. So what I would like to do is to acknowledge my wife. Sorry, the slide's disappearing at the top of the page. I acknowledge my wife's fundraising exercise for the Rare But Real walk we attended yesterday. She cajoled her friends, neighbours and work colleagues and managed to raise a good bit of money. It's actually slightly higher than that. It's more like 2,600 now. I also wanted to thank my wife for her support throughout my 7-year sarcoma journey. Without her, I probably wouldn't be standing here today.
Good morning.